microglia are the immune cells of the brain. They play multiple roles in brain development and maintaining the brain in a healthy shape, involving synapse pruning and myelination. But one of their most important roles as immune cells is that they're constantly sensing the brain. And this is what you're seeing here when they extend their processes and sense the brain environment. This is in vivo microglia. When an injury occurred, and what you'll see on this video is a green dot appearing here in the middle. This is a laser injury in the mouse brain. Microglia are going towards it. And by going towards it, they will phagocytose, engulf some of the cell debris or protein debris that have been released, as well as secrete a lot of cytokines to promote repair. When microglia are going from this phase of sensing to responding to an injury, they acquired very distinct transcriptional profiles that we name states. And with recent advances in single-cell RNA sequencing, multiple microglia states have been identified in mouse. And this is just an example of some of them that have been identified in the mouse brain. We suspect that all these distinct microglia states have specific functions and unique functions in the brain, but that's something that still remains to be confirmed and studied. We also know that there's a lot more diversity in terms of microglia during development, and that diversity goes down in adulthood and then restart in aging. So as you can imagine, there's a lot of microglia diversity in critical stages of development and aging that might be linked to diseases. And that's why my lab is interested in understanding what is driving the response of microglia? What is driving microglia going from a homeostatic sensing state to some of those distinct state that have a unique transcriptional signature? And to put that in the disease context, we want to understand how the genetics of those diseases impact the transitions, as well as how the brain environment, the brain regions, the pathology this is happening is also affecting the transition into specific microglia states. So that overall, we can understand how these states are produced, and we can identify which ones are protective, which ones would benefit the brain and would help delay disease progressions, so that we can identify means of promoting those protective microglia states in the context of disease. The diseases where we know most about microglia is Alzheimer's disease. It's been identified now for over almost 10 years that there's a distinct microglia state that is found in the Alzheimer's brain called disease-associated microglia. We can see those microglia around the amyloid plaque, which is the hallmark of AD pathology. And these microglia also express a lot of Alzheimer's disease risk genes, such as TREM2, suggesting they might have a role in disease progression. But we know that there's a lot of aspects of disease that is affecting microglia. Not only there's a lot of risk genes that are highly expressed in these cells. Amyloid, as I mentioned, attracts them, but also aging. Normal aging will induce microglia diversity. The brain environment, different brain regions, and different phenomena happening in the brain will impact microglia. And finally, any brain-body interactions will also impact microglia. So it's really complex to understand exactly how the disease-associated state is induced in patients and what is their roles. So today I'll show you two different stories. One about mainly the tools that we have developed during my postdoc to study microglia states using stem cell models. And I'll talk about some of the basic, I think the paper that I referred in the talk abstract, and how we can study these states in vitro, which makes it much easier to study them and grow enough cells to do studies than in vivo. And then I'll continue into how we can understand what's happening in disease brains by having a study on lipid toxicity. For both of these parts of the talk, there'll be some villages or pooled culture and multiple lines. So we'll get there in both parts, but I will both start with a single cell line first. So one of the challenges that we had, as I mentioned, is to know what is happening in disease and what is inducing those disease-associated microglia. We know that in mouse studies, as well as patients, so many things are changing in the brain. And we thought that if we could recreate these states in vitro, it would really help us to understand how microglia are changing and what is inducing them. So we treated our iPS-derived microglia with what they usually see in the disease brain, either apoptotic neurons, amyloid fibrils, myelin, or synaptosomes, which we know are all changing throughout disease progression. The cells were treated for 24 hours, and then we sent them for single-cell RNA sequencing. The first thing that we could see on the UMAP at the bottom was that when untreated, the cells on the UMAP are colored in purple or very homogeneous, which is something other groups have studied, and that's why it was thought for a long time that microglia could not really be studied in vitro. But what we saw is that when the cells are responding to one of the substrates, so the cells in gray on the UMAP, they're much more heterogeneous. And when looking at the transcriptional signature that appears, we could see some of our favorite microglia states that have been identified in vivo, such as the homeostatic microglia, proliferative, interferon, antigen presenting, and finally the disease-associated microglia that has been identified in Alzheimer's disease patients. As you all know, these UMAPs are very biased on looking at our favorite genes. And we got very excited, but we wanted to have an unbiased way to make sure that the cells we were seeing in vitro were very similar to what is happening in patients. So to do this, we teamed up with Evan Michalsko's group, who has an amazing microglia brain bank, and where they've sequenced over 80 patients to this day. And we could see that by doing LIGO, we could align our IMGL data with their brain biopsy data very well. And that the disease-associated microglia, which come here as cluster 4, was extremely similar across the human the human in vivo as well as their IMGL models. We have done the same exercise with a lot of other human data sets with mouse Alzheimer's disease model, the xenograph model, as well as confirm some of the findings that were previously known about disease-associated microglia with our specific signature. All of this to really confirm that what we were seeing in vitro was valid and representative of what the field knew so far on disease-associated signature. However, there's still a lot of questions to be answered about these disease signature. And the first one is what is regulating it? So we've known for a long time that the signal probably starts from the TREM2 cell surface receptors, and it would lead to the disease transcriptional signature. But what was in between is still largely unknown. We decided to answer this question to focus on transcription factors. And to do that, we combined two different analyses. One was a bulk ATAC-seq, where we could identify changes in chromatin regions that were changing in response to apoptotic cells, and from this identify transcription factors that might be more active in these conditions. This is a bulk method, so we knew from the diversity of our signature that we would not pinpoint specifically the disease-associated transcription factors, but we would be able to see them among others. And the other method we've done is a scenic analysis based on the single-cell RNA sequencing data that we had. This is an analysis that is based on the RNAs that are expressed in the states we were interested in. So it's a predictive model, but it's a single-cell analysis, so that might be able to get us closer to the disease signature. From these two models, we identified transcription factors, and 17 of them were actually common across the two analyses. And one of them that was very interesting to us was MIDF. MIDF is highly expressed in the disease-associated signature. And when we started looking into other publications, we've realized a lot of groups have identified MIDF, changing in mouse, changing in human patients, but no one had really picked up on it because it's usually not expressed at all in the brain. So we have decided that we would test if MIDF is really a real generator or transcription factors involved in the disease signature. But to do that, we had another challenge to overcome with microglia, which some of you might have encountered also while working with microglia, is that they're really difficult to genetically manipulate. So microglia are resistant to transfection, electroporation, and any sort of viral infection. So we've worked with Ben Deverman's group, and I've identified that if we add a VPX particle in our mix with the lentivirus, we could boost the virus infectivity from about 5% all the way to 85%. So we're not only manipulating 5% of the cells anymore, we're doing this for most of the cells, with it 85% is similar numbers that is achieved for most cells. We also wanted to make sure that the addition of the lentivirus and VPX was not affecting microglia too much. So we've done single cell RNA sequencing, and we really did not see much changes other than for some proliferative cells. But we know that some of the key microglia marker were very well conserved and were not affected by our lentivirus protocol. So that now we could finally test if MIDF would impact disease-associated signature. So to do this, we overexpress MIDF as well as mCherry as control and send that for RNA sequencing. When we look at the differentially expressed genes, we could find that most of them were related to disease-associated signature, including some of our favorite ones that are GPNMB and LPL. So that for the first time, we could genetically induce microglia states. And that was really important because in the past we could only observe them, but now that we can manipulate them, we can ask what is their function. And one important aspect that we needed to confirm is that what is their function on microglia phagocytosis? We know this is the main function of the cells in the brain. We were already at doubts that it was in the disease-associated signature were involved in phagocytosis, but by overexpressing MIDF construct, we were able to confirm that the sole overexpression increased phagocytosis pretty dramatically in the cells. So that's really the first kind of information we know about what would happen if in the future we were to modulate disease associated in disease. All these studies, as you imagine, are all were all end-stage studies. So we could identify microglia having the signature, but it was really difficult to know why were some cells were not acquiring the disease signature, and also what was happening after they had acquired the disease signature. So to answer this question, this is a newer tool that I've been working on here in my lab, is to develop reporter line when we can track the disease-associated signatures, and others we're working on at the moment. The first gene we have decided to track with those iPSC reporter line is ApoE. We know ApoE is a marker of the disease signature. It's an important risk gene in Alzheimer's. It's also, most importantly, a gene that is not expressed at all or very lowly expressed in homeostatic microglia, and the expression becomes much higher, very specifically in the disease signature. So to track the rapid transcription of ApoE, we added the destabilized GFP, which has a half-life of a few hours rather than the regular one. So it's much shorter, and we could track the cells that would express it. We have generated 2 clones that have the insertion of the destabilized GFP, 2 clones that don't have it. We've done some quality controls for pluripotencies, for expression of the ApoE expression, and microglia states. And we are now starting to ask some of the fun questions we can with this. And one of them is, when are the cells acquiring this? When are the cells starting to express ApoE? This video is slightly too fast. Cells are moving a lot. If you've worked with microglia, you know it's normal. Once we start treating them, they move a lot. I think for this one, we were taking 1 picture every 5 minutes, and it's still too fast. So we'll need to take more picture in the future. But we can track how many cells are expressing ApoE. And we can track also with these images, we can start looking into some specific microglia functions and look at morphology. One important finding we found using this reporter line was that the cells that acquire ApoE expression, so we see after 24 hours, about 20% of the cells acquire ApoE expression, which is very similar to what we were seeing from our single cell studies. But from those 20%, about half of them maintain the expression over 2 weeks. So we've been imaging the same cells exactly at the same location, the same wells over 2 weeks. I really was the first one to think that we would not see anything at that point. So we stopped imaging at 2 weeks, but we could really track the cells, and some of them were still very brightly positive 2 weeks later on, suggesting that there is diversity that's coming from genetics, but in this case, there's also diversity that's coming just among the cells that are in the same well. And some cells just seems to be more prone of maintaining ApoE expression. And right now, using cell painting, we're trying to understand what is making those cells special. Why do they maintain the transcription, the expression of ApoE for so long? We've also done a lot of experiments where we washed off completely the substrates of the cells. And so we're sure that the cells are not continually engulfing the substrates and they're really just maintaining it over time. And this is the first iteration of this reporter line, but we're also working with other reporters. We know that the disease-associated signature, similar to any transcriptional signature, is much more complex than a single gene. But as you might imagine, we are reaching a technical capacity to track multiple genes in the same line at the same time. But we are working on developing other markers, other reporters that can help us also understanding other aspects of the disease signature and of other transcriptional states. So that together, this first study was very useful to understand, to first develop tools where we could study microglia states in vitro. And not only we could identify them in vitro, we could study how we were inducing them with some of the specific brain challenges, but we could also assess what is their function by genetically manipulating them, where we've shown that the MIDF would induce the transcriptional signature, and that would lead to an increase in phagocytosis. While we've done this study, most of the study has been done in H1 embryonic cell lines. We have validated some of these findings also in 3 additional healthy donor cell line. It was still a pretty small number. And it's clearly the kind of numbers that will not get us into understanding how genetics of Alzheimer's disease impact these cells. So as a quick summary on I'm sure you all know this, but a quick summary on Alzheimer's disease genetics, why we were so interested about Alzheimer's and looking how the genetics impacts microglia is because a lot of the risk genes that have been identified are actually highly expressed in immune cells. But that's basically the only similarities across these. We know that they play a lot of different roles in the cells. So it's really difficult. It was difficult to pinpoint exactly how they would impact microglia, if they would all impact microglia in the same way, and if there was a common pathways that would come up in pathologies. To do this, we've encountered the same problem as I'm sure many of you have encountered, is how could we scale up some of the cell lines? I will go quickly over the next few slides because I think you've seen this and you're aware of this. If you have questions, feel free to stop me about the demultiplexing of the lines. But we've been using Michael Wells' method. We've been working together when we were both at the Broad, and we've adapted the cell villages for microglia. He has been mainly working with stem cells and neural progenitors and neurons. Microglia had a unique feature where they do proliferate throughout the differentiations, and that added an additional challenge to applying this method to the cells. But as you'll see later on, we've been able to handle this decently well. Another aspect that we've done is to adapt it to look into some of the main features that we're interested in microglia, which were mainly microglia states. So using the single cells RNA sequencing approach, we could look at the various microglia states that have been identified. And using the census-seq approach, we've been focusing on microglia functions. So that from the same villages, often we could look into proliferation, phagocytosis, and sensitivity to different stressor, as well as having the matching single cell transcriptional states values. So that has been extremely beneficial so that we can match and link microglia transcriptional states to functions. We have been lucky in a way that microglia can be easily isolated. So we've been able to adapt this, and I'll show you some examples throughout the talk of this, not only for microglia monoculture, but also for co-culture, slice culture, and the chimeric models, so that we can inject multiple cell lines in the same animals and then do basically the same assays as we can do in vitro all the way in vivo. And finish with some example of how we're using that at the moment. To select our lines, we've been working with the CERM Biobank. And in our case, it's because at the time, we really wanted to have access to the largest number of cell lines available. And in that, that was the only one that was available when we started this. And we wanted this because we actually did not want to focus on a specific Alzheimer's disease risk genes. We wanted to have a broader understanding of what is happening in patients. So we calculated a new Alzheimer's disease polygenic risk score. We applied it on the CERN biobank, so the full 3,000 cell lines, and we selected the lines that had that were at the extreme of our distributions. So that these lines sometimes were not Alzheimer's disease patients, but they did have an high risk of developing Alzheimer's based on this polygenic risk score. We have tested the polygenic risk score across some of the Alzheimer's disease biobank and data we had, as well as some of the CERM Alzheimer's disease patients. You might know that CERM has a small Alzheimer's specific biobank. These numbers and this specific graph was done a few years ago. I know they have expanded it a lot more, but from these numbers, we could see that if we compared the 70 Alzheimer's disease patient they had at the time in pink, they had a higher polygenic risk score than if we compared to the donors that were assessed for cognitive decline and didn't have any. Those numbers are small, but it's something that we're redoing at the moment with their newest cell lines available. So using the CERN biobank, this is all the line, each dot represent a donor, and we've picked the ones that are colored in pink. So we've picked the ones that were the extreme polygenic risk score, as well as some lines that were at a 0, so a middle polygenic risk score. We went independently of ApoE. ApoE is one of the main risk factor for Alzheimer's disease, and we've decided to go independently from ApoE to select the lines. We know this is something that will affect the results. It is always a covariate that we take in our analysis. I have to say at the beginning, we only had SNP genotyping to calculate the polygenic risk score. And the ApoE genotype was accurate in about like 75 to 80% of the cases. So we just didn't want to discard cells that we had just bought because they didn't have the ApoE that we have just that we thought. So that's why we decided to go independently of ApoE, but use that as a covariate. One of the first questions we had, and the same worry I'm sure you're all having, is what's going to happen when you put cells that are sensing each other all in a dish. Well, that's why we did this pilot study with 27 lines where we pulled the cells together, we differentiated them, throughout the differentiation, and then treated them with this is treated with apoptotic neurons only to see if we could observe some of the same disease-associated microglia. Oops, there's a little problem with the animation here, but you can see from the UMAP on the left that the donors are pretty well distributed across the different clusters. And when we look at the signature of each of these clusters, we could see some of our favorite disease-associated microglia, interferon, and proliferating microglia states. We aligned the signature with the human biopsy data set from Evan McCusker, similarly to what we've done in our previous study, again, to be very unbiased. We do see two clusters that are mainly driven by either the in vivo or the village data, but for most of the other ones, they're pretty equally distributed, especially for those microglia states that we're interested in. So that was a good validation. I did mention that microglia proliferate. If you've worked with them before, you know that proliferation can vary a lot. And we've had some disaster villages happen where one line took over. In the case of the CERM line, I think this has really been helpful for us that the cells were proliferating relatively similarly across the CERM lines. We have been encountering more problems when we combine cell lines from different biobanks that were produced differently. But as you can see here, this is the distribution of the same 27 village 27 donor village that I presented earlier. And we collected the village at pooling at day 30 and day 40 at the end of the differentiation. And across the villages, it stays relatively the same. And most importantly, there's no one line that is taking over the full village. So we can have a good representation of the cell lines at day 40 in our final assay. Using the data from proliferation as a proof of principle of what we could learn, we have calculated a phenotype score for proliferation. Basically, if the score is above 0, the proportion of cells increase. If it's below 0, it decreases. And each donor was assigned a score. And you can see that most of the score across the 3 villages are very similar. And that was just a way to quantify what we were saying and not just basing it on the graphs that I shown you earlier. So from these scores, we're also curious to know what is driving the proliferation. Microglia proliferation is something we don't know much about. So we've done a QTL analysis. And we don't reach whole genome significance in this case. We do not reach in any of our assays at 27 cell lines the whole genome significance. But when looking at specific hits, we obtain variants that were in genes that have been linked to myeloid proliferation and HDAC4, which is an inhibitor of proliferation. So we're getting some biologically relevant hits, and that was reassuring. The other microglia functions that we were interested in doing is phagocytosis. It is one of the main functions of these cells in vivo, and it's important to know what is regulating these microglia phagocytosis. We can track phagocytosis by feeding microglia, which substrate for phagocytosis that is linked to pH rhoda. So the cells will become fluorescent when it is engulfing the substrates and when the substrate is in the low pH of the lysosome. Using low flow cytometry, we can isolate the cells that have high phagocytosis, so high pH rhodo signal, and others that have low pH rhodo signal. We've sent that for census-seq analysis. You can see here the pie chart and distribution. And we can see already that from this, like the olive donor is much more represented in the low phagocytosis than it is in the high. So that would be considered a low engulfer in this situation and in this village. Using the same approach to quantify as we've done for proliferation, we've given a score to all our donors. Here again, you can see the scores are very consistent across independent villages, except for this one, that this seems to be more variability, but for most of them, they're very consistent. The low engulfer are low engulfers across villages, and the high are consistently coming up high. For this, we were interested in knowing how we could link it to genetics. And I've talked a little bit about our polygenic risk score. Well, what we could see is that the donors that had a high polygenic risk score, so a higher genetic risk of developing Alzheimer's, had low phagocytosis. You can see there's still a lot of variability around the 0 or the middle ones. It's something we're interested in looking. It's clearly a limitation of our study with only 27 cell lines. In this case, we cannot have a broad understanding of what's happening at both tail and in the middle. But there's a lot of variability still that exists, and we're still very intrigued into where is that from and why some donors seems to be engulfing so much more, and it might be something that is independent of this polygenic risk score. We have also done a QTL study on the same data. And what we can find in this case is that the top hit is TMEM106B. TMEM106B is a lysosomal protein. So it's something that is known to be involved in phagocytosis, and it makes a lot of sense to be coming up. Again, we are not reaching whole genome significance, but biologically, it makes a lot of sense that a lysosomal gene is coming up as a modifier of phagocytosis. So overall, I think what I've shown you so far is really that we've been able to develop tools where we can manipulate microglia states in vitro. We've been mainly focusing for these first studies on disease-associated microglia because of their roles in Alzheimer's, because there's so much other study that we can compare our result to, and I think that was extremely important in setting up the models. But we know there's a lot of other microglia states that exist that we can study with our assays, and that will be interesting to do very similar sets of experiment on to understanding how these states are regulated. But to go back to the initial premise of how do we understand Alzheimer's disease? How can we put it all together? We have the tools now, we have the cell lines that were selected based on a genetic score. Well, we had to find also in which environment are they changing? How are they impacted by the disease environment? And in recent years, there's been a lot of studies that have started highlighting the importance of lipid metabolism. We know also that many of those risk genes in purple are not only involved in immune response, but also in lipid sensing and lipid metabolism. So we have decided to look more specifically into that. We know, yeah, as I mentioned, that a lot of the risk genes are involved in lipids metabolism. Amyloid, interestingly, can trigger some of the same cell surface receptor as lipid receptors on the surface of microglia. There's been multiple studies that have identified changes in plasma and CSF composition of patients where they were altered lipids. And there's conditions that affects the body that makes individuals more at risk of developing Alzheimer's, such as diabetes, which is known to be involved in affect lipid metabolisms in general. But there's still a lot of questions that remains, like where does it start? Where does lipid metabolism start? And which cell type does it affect? But also, what type of lipid are we talking about? Lipid, as you all know, is a very broad category of molecule. And do all lipids affect microglia the same way, or is there specificity? Was something we were interested in studying. And we thought we had the appropriate tool to ask these questions. We have decided to focus on free fatty acids. So we know that fatty acids are essential in the brain. They're involved in myelination, in membrane remodeling, in receptor signaling, and they are found and essential for most cell types in the brain. There's also been recent studies that suggest that neurons and astrocytes under stress conditions will secrete fatty acids, and that will impact that could impact microglia. So we have decided in an unbiased approach to test how fatty acids were impacting microglia. And to do this, we teamed up with Anna Greca's group at the Broad using their platform called Falcon to study in an unbiased way free fatty acids. We treated our microglia with 48 different free fatty acids, and then we looked into the microglia states that were produced, their plasticity, and some genetic modifiers. What we could see when we looked at the transcriptional states that were produced, this was bulk RNA-seq, was that the impact of those fatty acids was very diverse across our microglia. And we could actually reorganize our cluster of fatty acids into 5 different subgroups that you can see here based on the gene expression. These subgroups actually shared some similarity at the level of gene expression. There's about like 60, 70 genes that are the same across the 5 groups, but most of them are extremely diverse in microglia. And this was surprising because we're not talking about the whole class of all lipids found in the brain. We're really focusing on fatty acids. So, but they still seems to induce at the same concentration and same time point a lot of change in the transcriptional expression of microglia. When looking at some of the pathways that were altered, we could see that many of them, not surprisingly, are almost as a positive control lead to changes in lipid metabolism. But when looking into distinct pathways is really when we started seeing the difference across the cluster, where, for example, cluster 2 is highly involved or induces an immune response in microglia. Cluster 3 is inducing a lot of signaling pathways in microglia. And then cluster 4 is involved in protein processing and cluster 5 in metabolism. So they all induce very unique pathways. And our first thought was that maybe it was an organization of the fatty acid based on their length or on the type of fatty acids that we could identify. But we saw that some clusters specifically, like for example, cluster 1 is really a combination of saturated and monounsaturated fatty acids. Cluster 3 was also a mix of a lot of the common fatty acids. So it was something that was a bit deeper than just the type of fatty acids that the cells were responding to. To continue further, as you might imagine, we could not continue testing those 48 fatty acid constantly in our assays. So we've selected a few of them to do some of the follow-up characterization. But the few we selected had very similar gene expression profile as the group of fatty acids. We went on to validate this signature across multiple iPSC lines. So we have, in this case, 3 different iPSC lines that had similar signature. We've done dose response and time course. And I'll talk to you a bit more about some of the functional impact we've seen on these from these fatty acids. And for this, we've been mainly focusing on cluster 2 and cluster 3. If you see cluster 2 as microglia are responding, but their morphology is relatively similar throughout time. I stopped this after 6 hours, but there's multiple other pictures, and they have a normal morphology. While cluster 3, as you can see, the microglia morphology is drastically altered. They lose completely their processes, and they completely stop moving. And when we've looked, we can see this very early on after the treatment of the fatty acids with microglia. And early on, as well as later, we see the very similar signature in transcriptional changes. So these changes are happening early and very early on affect the cell's states and functions. What was interesting in this is that not only there was a change in morphology, there was a change in lipid processing. Cluster 2 cells accumulated lipid droplets that we stained with PLIN2 or body P, and as we would expect when we treat the cells with any sort of lipids. However, cluster 3 cells were very resistant. We have never seen staining, or we've seen very low staining of PLIN2 or body P, suggesting that the lipids, the fatty acids, are impacting microglia, but they're not found within the microglia cytoplasm. And this is the quantification you can see on the right. And when we've looked at the toxicity, we had completely the opposite. So the cells in cluster 3 were highly toxic. By 24 hours, we've seen a lot of cell death as measured by propionine iodine, while there was very little cell death in the other ones, suggesting that it's not the presence of lipids in the cytoplasm itself might not be causing the toxicity. It's really the response of the cells and how the cells can process these lipids that is impacting microglia function, and in this case, toxicity. Again, we've done this across different time points and different concentration. And an interesting finding is that we do not see differences when we treat neurons. So it's something that the cluster 3 toxicity is specific to microglia and has not been observed in iPSC-derived neuron models. One of our first guests to know how this toxicity was modulated is through TREM2. I've talked that TREM2 is the main cell surface receptor for the disease signature, and we know it is a lipid sensing molecules. So we decided to take TREM2 knockout cell lines and treat them with our cluster 3 fatty acids. But we did not see any change in toxicity, as well as any change into the transcriptional signature, which was interesting. I think for a long time, TREM2 was the answer to many questions in the field of microglia and Alzheimer's disease. In this case, we found a phenotype that is not modulated by TREM2, but it still left us puzzling into what would modulate this transcriptional signature and the toxicity. And that's where we turn to our village approach. Instead of going one gene at the time, I've done a few of them. I got disappointed very quickly, and I think my guessing game was just not good. So I've decided to take the unbiased approach and go with cell villages to identify new modifiers of the toxicity we're seeing. So in this case, the cells were pulled together, differentiated, and then we treated them with our cluster 3 fatty acids and identified the donors that were viable through flow cytometry at the end of the experiment. We ran QTL analysis to identify which variants was affecting the response to this toxicity. We found when we don't have, again, any whole genome significance results, even though some of them are getting pretty close to in this case. But taking the top variants, we've identified over 400 variants that were in 300 genes. So most of them were in coding regions. And not only that, when we could look into where in which type of genes they are expressed, these variants were mainly found in microglia. So this is a collaboration with Chris Klass and Joanna Czyklasky at UCSD, and we could see that most of the variants we have identified are microglia, which was extremely reassuring. This is something that it's difficult, or we don't have a lot of proof of principle of genes that would affect fatty acid toxicity like we had for the proliferation or for phagocytosis in our previous QTL studies. But just knowing at least that what we are finding is very unique and affecting genes in microglia was a good first pass. And right now we are targeting some of these genes using CRISPR to knock them down and see individually how they impact microglia's response. And we're also working on the analysis on this results to see how ApoE genotype is affecting the phenotype, as well as assessing the sex difference, which I'll get back later on. So together with the studies, we were able to use a platform we have developed earlier and start answering some questions about the role of lipid in the context of Alzheimer's disease. We see that the microglia respond very differently to different types of lipids, and that the lipids will impact at the same time the transcriptional states as well as the function of these cells. But there's still a lot of question that needs to be answered. One that we are working at the moment is that we know lipids and changes in lipid metabolism is a common pathological sign across neurodegeneration. It's also been observed in amyotrophic lateral sclerosis, and it's something at the moment that my lab is trying to compare. Are the same lipids and the same rich genes involved in both of these diseases and affecting the disease progression? And another one that we're still working on to answer is what does trigger lipid changes? Now with in vitro, we can treat the cells with distinct perturbations, but in the context of disease progression, where do these changes in lipid comes from? Is it from the diet? Is it from lipid processing in the brain? And to get to this answer, we had to go a little beyond the in vitro models because we just don't know when it's happening, so we don't know what to treat ourselves with to recreate this this phenotype. So that's when we started working with microglia villages in vivo. We're using the xenograft model that was published by Matthew Blurton Jones' group at UC Irvine, where you can inject human microglia in the mouse brain. We've been able to inject villages completely in the same animal. So the cells are injected around 3 or 4 days after birth, and then we can let the animal age. We can very easily isolate microglia from the mouse brain and isolate the human one using human-specific markers and sort them through flow cytometry to do single-cell RNA-seq or a different census-seq village. As a pilot, we've injected 1, 2, 3, 4, 5, 6, 7 SN lines. I should have written this down. And we can see that, yes, the cells engraft differently. That's something that was reported. Not all donors will engraft in the same way. But across the cells that were collected, this is across 3 independent animals, the engraftment is very similar. And again, we've quantified this and to look at the engraftment score, and it's very similar across animals. So we went on and decided to inject 40 lines into the same animals. And what we've done is we injected at day 3, let the animal age for 6 months, and then treated the animals with LPS. We know lipopolysaccharide will induce pretty drastic changes in transcriptional states in microglia. And we were curious to know if the genetics of these cells could highlight the mechanism by which LPS is impacting microglia. You can see here from the cells that were collected, the distribution looks fairly similar across the two conditions. There was 4 animals that were treated with LPS, 3 animals that were treated with saline. And then using single cell RNA sequencing, what we could see is that the donors are distributed across all the cluster that we have observed. And as you can see here, the LPS treatment induced a pretty massive changes in the gene expression. We were expecting something pretty drastic. We see a lot of genes that have been previously identified, such as IL-10 changing, but really we have pretty distinct clusters in this experiment. So that was interesting and something we are still working on the analysis at the moment. The last slide I want to show is a study we are doing at the moment and something we haven't talked much so far in this talk, but all the cell lines I was using so far were male cell lines. We know that in the context of Alzheimer's disease, women are twice more affected than men. And that's something that we still don't have all the answers into understanding. And so we have decided with the support of the Alzheimer Association to develop a study where we would look into how female and male donors with ApoE4 variants alter microglia states. We know that ApoE4 across women is increasing the risk, but also accelerating disease progression. So in this village, we are pooling male and female donor lines. They will be all in the same animals. Animals will be male and female, and we will isolate these cells. And through single cell RNA sequencing, we'll be able to identify how does the sex of the animal impact microglia states, and also how does the sex of the donors impact microglia states that are produced, and then apply our polygenic risk score and see how the polygenic risk score can have a synergy with the ApoE4 genotypes. So that's a study that is ongoing right now, and we're really excited to get going on this and hopefully broaden our understanding of what we know about microglia states in the context of disease. So overall today, I've covered some of the toolbox we have developed to study microglia states. Of course, these models and tools are constantly evolving, and they're not perfect, but we think that at the moment we have very decent tool to study microglia states in vitro, all the way, and also understand how they're changing in vivo. And we are also applying these tools to study lipid metabolisms in different contexts. And again, we don't have all the answers, but we know that microglia are very unique sensitivity to some specific fatty acids. And we've identified some modifier of this, and we're very eager to test how this will impact disease progression. So I will finish this. I'll finish by thanking my amazing lab. I started my lab a year ago. I think you need very courageous people to join a new lab and be ready to unpack boxes and reorganize everything, I feel, every few months. So I'm very thankful to all of them, as well as my amazing collaborator and the support from the funding agencies. So thank you. I'd be happy to take any questions.
Thank you. Thank you, Martine. Jump in with any questions, please.
Can I ask a question? That was just incredible talk, so clear. Thank you. Thank you, Martine. So I wanted to ask about the specific fatty acids that were so toxic. Are those ones, are they increased in CSF or in body fluids with disease or inflammation?
Yeah, I wish it was a simpler story. I tried to go quickly on the part of the CSF studies because techniques like detection of the lipids varies a lot, and not all type of lipidomic will detect fatty acids. And also a lot of this the number of patients in these studies also varies greatly. So that's why I'm always cautious on making strong conclusion about it because it varies a lot. Yes, we have found some studies that suggest they're changing, but sometimes they're not found in other studies because the other study didn't use a method to detect fatty acids. So, and then sometimes the study is just as powered the way they could with a small number of patients. So that's why I'm very cautious about making this
Sure.
conclusion.
Sure. Yeah. But are they related to the ones that Shane has found?
Yes, we are
Yes.
working with
Oh,
Shane to test
awesome.
that in astrocytes. It's something we're very interested in putting it all together and seeing if, if astrocytes are responding similarly. But yes, we are working with Shane on this.
Awesome. Great. Thank you.
Thanks.
Can I ask you a question? A couple of very general questions. So one is, when you model microglia this way using iPS cells, how does this interact with the sort of uh, hematopoietic stem cell derived macrophage microglia and the endogenous microglia story? Are you getting specific kinds of microglia this way?
In the
Do we know?
xenograft or in vitro?
Well, let's start with in vitro.
Yes. So some of the markers we see, and that was always a kind of caveats that was raised in previous studies, is that microglia in vitro would lose their distinct microglia signature. But what we saw is that the signature comes when the cells are responding to the different brain substrates, so to myelin, and we think it's really like it's part of their environment. It's their ontogeny, but it's also part of the environment that pushed the signature of microglia to become microglia and not macrophage.
Oh, great. So that's sort of segue to the next thing I wanted to ask you. So when you talk about Alzheimer's and all this variation, and you talked about polygene risk scores a bit, uh, can you classify, does the data that you have on the Alzheimer's donors allow you to classify by BRAC stage so that you can really assign a kind of developmental risk score as well as not risk actually, it's a developmental clinical score alongside polygenic risk? I mean, I said these are very naive, so I'm not,
Yeah, no, and there are other studies that have shown a link between the polygenic risk score with BRAC stage. In our case, the polygenic risk score we have not compared it to a biobank where we could have all the way to the level of pathology. We had the genetics, we had the diagnosis, but we did not have pathology in the brain like BRAC stage, so we could not link it there. It's something that we know the polygenic risk score we calculated is already a few years old, and now there's been new studies, so we are updating it, but I don't think we have the data also for the BRAC state. It's something that would be really interesting, and that probably could be done with the ROSMAP study where they have brain pathology, genetics, and iPSC. But in our case, because we've used CERM, we don't have that information altogether for the lines that I've been working on.
okay, there's tons of questions. Thank you very
I
much.
Yes, it
Yeah.
would be great to have the full spectrum together and have all this information for sure.
Maybe I can just ask one more. What about tau? So you talked about ApoE, but another major risk
Yeah.
constellation for neurodegeneration is the tau system.
Yeah. And it clearly plays a role. I think we can have a full hour discussion of amyloid versus tau, and how tau is probably more linked to pathology. I have done a study during my postdoc where we treated the microglia with human tau as well as human amyloid. So the amyloid I use in the case of the initial study is fibrils, so it's a laboratory-made like peptides. But we have selected. Sadly, though, the two times I did it, we got really unlucky and the single cell sequencing didn't work. sadly. It was a very tough experiment. But so we don't have the answer for that and how microglia are responding to tau. But yes, it's the kind of question that we can answer now with the platform. We could also compare across protein aggregates, across neurodegeneration.
Right.
That would be very interesting to see if the cells just respond similarly to protein aggregates or if they have unique signature across disease.
Cheers. Thanks so much.
Thank you.
I have kind of a general question on strategy or experimental design when you're trying to look at variation across humans. I don't know if I can remember the exact setup of the lines you use for the in vivo engraftment, but how do you in general think about the value of adding new lines versus adding replicates of lines? I know you also mentioned the fact that if you derive a line a different way, that can cause a very large difference in proliferative qualities early on, which I think kind of terrifies all of us, right? Just that the way it was derived would matter so much. Yeah, so maybe your general comments on any of those points.
Yeah. Adding more lines where I think especially in for all these contexts, the replicates is really important. Like especially in the context of mouse work, the xenograph one, we know there's so much variability that can be seen across animals. And that's something that's really important. But the advantage of having all the cell lines in the same animal is that we can see and observe if there's a differences. And something to keep in mind for those in vivo study is the type of perturbation. So we have first tested LPS because we know microglia change drastically. And we don't know at the
That was
moment.
peripheral LPS, but a central assay, is that correct?
Yes, IP injection, yeah, peripheral LPS.
Yeah.
And it's because we don't know at the moment how microglia are engrafting. Is it going to be like one donor that goes in the little region of the brain and then proliferate and basically have a lot one cell lines from mainly one donor in one region and then another one will be in another region, or already constantly moving across the brain? Like it's something that's it's very technically challenging to know who is grafting where in the brain. And I'm just saying in the brain, but we know they all go all the way down to the spinal cord. So who is engrafting where is a question. So that's why we went for LPS, because we knew that most microglia across the brain would respond rather than a very unique change. And that's something that, yeah, that's for completely another set of experiments to know if we do like a very precise change in the mouse brain, which microglia will be changing, and are they all going to change? So that's something I would be mindful of in designing the xenograph experiment to make sure that all the cells will be impacted, because it's not because one cell will not respond that it means it doesn't respond to the challenge. If it's not in the right region of the brain, it might also just not be responding because of that.
So you were mentioning replicates of assays and having a lot of things happening in the same animal. Have you ever incorporated replicates of multiple lines from the same donor? So multiple reprogramming events from the same genome in any of these systems?
That I have not done.
Okay.
I have not done. We've taken all our lines from CERNs. I know some of them we had access, they sent us one clone versus another. I have not done different ones. That was also one of the limitations, or like the first or advantage of setting up this project is that we had access to CERN lines. So that kind of made it easier to just select the CERN biobank, and we knew they were all produced the same way. I think we've had mainly issues when we tried to add isogenic lines. Like in the best case, like originally when we designed the experiment, we would put a TREM2 knockout lines that a lot of group has studied, that a lot of the group have clear phenotypes, so we can almost like benchmark our assay based on that TREM2 knockout lines.
Having that as one of the lines in your village, you mean?
Yes, so
Very
adding
cool.
this.
Very cool. Yeah.
Yeah. However, that line just took over. And we've realized the more the lines are genetically modified, and if you like, it's the line from Coriell that has been genetically modified. So just the iPSC is growing so much faster than everything else that sadly it took over the village. And that's
That's a wonderful addition to an experimental design to have a built-in,
yes.
like you said, baseline or reference, not baseline, but effect reference almost.
Yeah, so we have not been able to do that yet. It's something we're still working on, maybe generating like mutations already in the lines we have. But I think I've been very mindful of keeping the lines that have not been genetically modified or manipulated too much or grown to a level that is needed for this, because clearly if they start proliferating too much, it will impact the village.
Gotcha. Thanks. Ron, did
You
you
know,
have another question?
we've had a talk we've had a talk before about this issue about IPS lines growing differently. You might want to go and look at the video. It was extremely interesting because Carlo, you may remember this better than I do, but one of the results was a human population genetics result suggesting that different SNPs were marking the expansion of cells at different stages in the expansion, so that the expansion is not just one kind of mess, but it's actually a series of precise proliferative controls that are coming into play at different points. And when you say one line took over, that's going to be P53.
Yeah.
Again, we've had talks about that. So, and of course, that's all completely relevant to this issue, this astonishing thing that you can repopulate the brain with exogenous microglia, right? I mean,
Yeah.
I have this correct, don't I?
Yes.
That's mind-boggling, really, that you can do that. Yeah. Okay. Peace.
Yes. Yeah, no, I think you're right. It's probably the line that took over probably at like a P53 mutation. It would be interesting to track. Yeah, maybe it'll do looking for variants at different stages of the differentiation could help us understand and see what is happening there. If it's how biology is driving this difference. But yeah, so far it's been with the SERM lines, we can keep a decent their population under check a little bit, and no line is completely taking over. And it's been very helpful for these assays.
Maybe I can talk for a minute about something that I've always found interesting about microglial diversity, which is it the case that if you sample microglia from the fetal brain, the diversity is higher? And then in the adult, what happens is that the diversity reappears in response to particular challenges, as if the fetal system is actually initially very diverse, and then it kind of focuses down, but you have this potential left, and you open it up. But it's like an immune system in a way.
It is, yes.
Well, it is an immune system.
Yeah, it is exactly what we're seeing. The diversity is very diverse in development as well as in aging. And I think the more we sequence, like microglia only represent 5% of brain cells. So they're often forgotten in those single cell studies. So that's why we really need to enrich for microglia when we do those single cell study to make sure we capture the whole diversity.
I see.
And then also they have in our mind, like in our hands, they have like a slightly higher mitochondrial rates. So the usual single cell cutoff, you can cut off a lot of your microglia. So I would suggest if anyone has data to look into that, maybe you can get your microglia back in. But we need so many cells from a patient or from a single animal to see the diversity. But once it's done well, we can see the microglia diversity in development as well as aging. And I think we're just going to learn more about this diversity over time. Something that is interesting also is that we're still narrow down the exact signature. We know there's a lot of species differences. So between mouse and human, there are a lot of differences at the level of the genes that are markers for unique microglia states. So we need to really do the experiment in the right species. But some signature looks similar to the ones we see in aging are kind of similar to the one we see in development. So could they be like reactivated? Is it exactly the same thing at the moment? We don't know, but clearly some markers are similar.
Thank you.
Thanks.
Coming back to the point you were making about proliferation, so many people do sort of make that graph that you made of what cell is taking over the culture. It's usually thought of as a baseline or a control or a setup experiment, but there's probably enough out there to do a GWAS just on proliferation, which would be interesting in itself to see the differences at those early times.
Yeah, now I'm curious to go back. I clearly still have, I could do that. I have the, I'll go
Well,
back.
yeah, I mean, for one, it was good because you didn't have big differences, but that might hurt your GWAS for looking for proliferation effects. But there was
Yes.
some in there, some
There were
differences.
some, we're not reaching whole genome, but there were a few top hits that were related. But now we did it at day comparing the pooling versus day 40. So now I'm curious to do one in the middle.
Interesting, interesting. Well, thanks so much.
Thank you for having me.
Really appreciate it. And yeah, hope to see you back in these sessions. Next month, we have Jason Stein coming in.
Great. I'll try to attend more regularly. Yeah.
Okay, everybody. Thank you so much, Martine. See you
Thank
guys next
you. Bye
month.
bye.